Hi, this is Miss Litton, and this is 7th period on a Friday, and these are some excellent scientists in the room. Can you please say hi? Hi. All right. So what I'm doing right here is doing mostly a review for your essay prep, which is how much of your test? 50%. And if you know that essay well, that should probably transition over well into your what? Multiple choice. But how would you really get ready for your multiple choice? Put yourself in the same situation as the exam, and that would be make yourself deal with multiple choice questions. Where could you get, they're tired, where could you get some multiple choice questions? Back of the chapter. How should you take it? Like as a discussion, flipping back and forth to the answers that are located in the back of the book? No, what should you do? Do it like a test. And at first I would do all the odds, right? If you haven't already done them. And I would have a piece of paper that I committed to. I would put myself in a situation similar to your testing environment. Go sit in isolation in a room. Don't have TV on. Put your phone away. Time yourself. Say, I've got 10 minutes to do the chapter 2. Or what are we on? What chapter are we on? 3, 4, 4 or 5. 4 or 5. Unit 2. Yeah. I've got 10 minutes to do the questions, or 15 minutes. That's what I would do. 15 minutes to answer all the odds for chapter 4, and then stop and grade it. Okay? Then do some reflection on that. And what did I miss? What do I need to go back and look at? You know? And then maybe then do a deep dive into the group shared notes. That's all input, right? Group shared notes. And then I would do the same thing for chapter 5. And I would do the same thing for chapter 40 that we learned about today. But did we discuss all of chapter 40? No. So are you going to hold yourself accountable for all of chapter 40? No. And I will not hold you accountable for the highly suggested reading and thinking yet for chapter 40. The only highly suggested reading and thinking you need to know is for chapter 4 and chapter 5. You got that? Just those 2 chapters, all right? Then once you've done that for both chapters and you've done some more input studying, I would probably, just lead with doing all the odds. Just see where you're at, you know what I mean? Get a little dipstick, check on yourself. Then I would do the same thing for chapter 5. Then I would do a deep dive into studying. And then I would do all of the even questions, like a test. Time yourself, whatever, 15 minutes. Check your answers in the back of the book. Okay, got that? All right. Now, next. Okay. Okay. Oh, crap. Sorry. Oh, bad word. My bad. I'm going to break these down. Do you have to madly type them? No, because I already gave you this presentation. If you want this presentation, it's in Google Classroom. Okay? And it's in the stream. So here are the big ticket items, and then I'll just kind of break them down one by one. You all right? Okay, and if you've made a copy, you can type right onto it. So the first one about your essays is cell organelles. I would be able to recognize any organelle if I gave you a picture. I would be able to describe the function of every organelle. You must differentiate between them. Like pair them up. Smooth ER, rough ER. How are they similar? How are they different? Mitochondria, chloroplast. How are they similar? How are they different? Make sure you can differentiate ER, Golgi. It'd be a really fun game, because I know that's what you're going to do. Go to Starbucks, put all the organelles in a hat. I pull one, my friend pulls one. I tell you what it looks like, what it does. They tell me what they look like. How are they similar? How are they different? Throw them back in the hat. Let's pull it out again. That is like fun times, right? Okay, no answer. All right, that would be one thing to do. Then one of your debriefs, a specific debrief. Okay. That was my robot voice I just went into. Then what ones, if I said, hey, what are the top 4 organelles? Why are these your top 4? Okay. Could you support that? That was one of your debriefs. Could you support why it is the most important, these top 3 are the most important? How could you do that? Top 4, top 5. How come, what fit in, what didn't fit into your top organelles, right? So explain, this is why these guys are in, these are why these guys are out. This is what I think is most critical. Does a cell need all of them? Yes. But I'm asking you to prioritize them based on their value, and in order to do that, you have to have a good understanding of that organelle. That's an application level. Does that make sense to you? I'd also want to know, which I did not put on there, which I wish I would have, is how did we get things like eukaryotic cells, right? The endosymbiotic hypothesis. Somebody remind me at the end to add that to this, okay? The endosymbiotic hypothesis. What does that state? Now, I haven't even written your essay test yet, but these are the things. I made a list of what I would ask you about, so I'm giving you the list of what I'm probably going to pull from. Could I pull something else? Sure. But these are the things I think are critical that are probably going to pop up on your test, okay? So endosymbiotic hypothesis. Should you know the difference between a plant, is this right? Okay, yes. Plant versus animal cell? How are they similar? How are they different? What kind of things would you see? Things like the energy organelles, mitochondria versus chloroplasts, right? Those would be things to look at. Who has the highest value? And the endosymbiotic hypothesis. And make sure you understand that. Give me the evidence for the endosymbiotic hypothesis if you were going to explain that to somebody. All right, moving on. Oh, and that stays. That's super cool. All right, so next, cell membranes, structure. So let's talk about that. If I asked you to draw a cell membrane, could you do that? A simple diagram, right? You'd be able to tell me what these are, and, oh, that's wrong. And if it's hydrophilic, hydrophobic. Okay. I would do a better job if this was my test. That's terrible. Okay, now tell me what else I need. I need some cholesterol. Your drawing should look like cholesterol. Cholesterol has how many rings to it? 4, because it's a steroid, right? See how this is inconsistent? Don't draw one like that. What else am I missing? Yes. And I would make enough room that I could put those on there. I wish I would have picked a different picture color. What did I draw right there? What was that? And this would be a glycolipid. Okay? Those are the 4 main components. Now, that's what needs to be there. If you wanted to add in something like this, what would that be? Cytoskeleton, right? But that's not one of the 4 components. You would make sure you knew, and you would know what each one does, right? That you could generally talk about phospholipids being a barrier, that you could talk about cell recognition and structure, that you could talk about the fluidity of the membrane with cholesterol, right? Those are the things. Proteins, there are several. What are all the functions of proteins? That's another thing you need to know. They could be what? Channels, carriers, cell recognition, receptors, enzymatic, and junctions, right? I will be able to tell what each of those do. So there's structure, there's protein functions, 3 ways to cross a membrane. What are the 3 ways? What are the 3 ways to cross it? Through the phospholipid bilayer, but not if you are large or charged. You could use a channel, or you could use a carrier, or you could do what? Whole membrane. You could be doing what? Endocytosis and exocytosis. But there's details on that, right? So when you talk about through the phospholipid bilayer, you could talk about what things can cross and why. When you talk about endo and exocytosis, you've got details on endocytosis. What are the 3 types of endocytosis? Phagocytosis, penocytosis, and receptor mediated, right? You know, I mean, you can dial down into that, yes? Okay, so structure, protein functions, 3 ways to cross. Oh, active versus passive. Active does what? Requires energy, and passive does not. What's an example of, could you name like 2 examples of passive and 2 examples of active? What would be an example of passive transport? What did you say? Diffusion of water across the membrane. That would be passive. And I heard another one. Facilitated transport. So you're going from a higher concentration to a lower concentration. That's what all passive has in common. A concentration gradient, a difference. A higher on one side and a lower on the other side. That's why passive works. Going from a higher concentration to a lower concentration. You could go through the phospholipid bilayer or use a channel or a carrier. That is passive. Okay? Active is going against the concentration gradient. Going from, I have only a few here and there's a lot out there, but I want to produce even more out there, and that requires energy. What are some examples? 2 examples of active transport. Sodium, potassium pump. We call proteins that use energy and concentrate a substance on one side, we call them a pump, right? So sodium, potassium pump pumps 3 sodiums out for every 2 potassiums in. Give me another example of active transport. Whole membrane. Any endocytosis, any exocytosis, your whole cytoskeleton is moving your membrane around. That's going to cost energy. Okay, got that? All right, any questions on active versus passive? All right, next. Cell membrane, osmosis, and the lab, and variations on the lab. So for osmosis, the key thing, what is the definition of osmosis? The diffusion of water across a semi-permeable membrane, right? Smelling my perfume, is that osmosis? No, why? There's no water, but there is water in the air, but what else? There's no membrane. There's no membrane. Okay, so in that one, you're going to need to make sure you remember, okay, you have some sort of membrane, and you have at least 2 solutions divided by that membrane. You could have more. Okay, if you have a bunch of stuff on this side, then that side would be referred to as what? Hypertonic, and this would be what? Right? Left and right. If one side's hyper, the other side has to be hypo, right? What else could I say about, this is side A, this is side B. What else could I say about it? High osmotic pressure. What else? High number of osmotically active substances. A high amount of what else? Solute. Okay? And this would be low osmotic pressure, low OASs, low amount of solute. Instead, it would have a high amount of what? Solvent. And this one would be low. You okay with that? Okay. So we know water must flow from the hypo. So it's going to go from side B to side A. So the water level should rise over time. Yes? Okay. So knowing and understanding that, we apply this to our lab. So our lab basically had 2 pre-labs, right? And then 1 main lab. So pre-lab number 1, okay? Pre-lab number 1, if I asked you to diagram or design pre-lab number 1, would you be able to show me a beaker and say you had a dialysis tubing, and what did we put inside the dialysis tubing? Starch and what? Glucose. And what did we put outside? Iodine. Okay? And if I asked you what moved and what evidence do you have that it moved, did iodine move from the beaker inside the dialysis tubing? Yes. What evidence do you have that iodine was able to move inside the dialysis bag? Change of color of the dialysis bag. What changed color? The starch, because the starch bound with the iodine, and then the iodine turned what color? Blue. That's my evidence that iodine could go through the dialysis tubing? Could starch go out of the dialysis tubing? No. Starch must have been too large for the holes. Remember, this dialysis tubing is just a model of a cell membrane for us to talk about how size matters, right? So starch was too big, it couldn't get out. If it was small enough to get out, what evidence would you see to say starch can come out? What would be the evidence? The whole beaker would turn blue. Did the beaker turn blue? No. So starch can't move. Okay? Did glucose move out? Did glucose move out? Yes. What's your evidence? Glucose test strip. So you had something you could test for glucose. It started out in the bag. It was still in the bag, but obviously some went out, and we could tell because we put a little stick in there, and we could test it for glucose, and it tested positive. So if I talked about what could move and what couldn't, I'm assuming water could move, yes? Okay, I'm assuming that starch could not move, but iodine could move and glucose could move, and I could give you evidence of that. How could I know if water moved into the beaker? I can mass it, right? Okay, so that was pre-lab number 1. Then we did pre-lab number 2. Pre-lab number 2. That scenario is you still had a beaker, right? And this beaker had what in it? It had distilled water in it. That just means double distilled water. It's very distilled, okay? Now, what did we put in those beakers? We put in dialysis tubing. Again, modeling a cell, okay? And what went inside the dialysis tubing? Different molarities, so questionable molarities of sucrose, yes? And the claim we all could make based on our curated data was as we increase the molarity of the solution within the dialysis tubing, then over time, if we set that tubing in water, what happened to the mass of that, the percent change in mass? It went up. So I could have given any of you during that lab a mystery solution. I could have had like 3 mystery solutions and handed them out. And if I said to you, find out the molarity of those mystery solutions, what would you have done to find out the molarity of those mystery solutions? Figure out if they gained or lost weight. So you would put, they're all going to gain weight, right, unless it was water, yes? But if you've drawn a graph, right, if we didn't graph this part of it, but if we had graphed, that's so beautiful. If this is 0, 0.2, 0.4, 0.6, etc., we would expect the mass to go up, yes? So if I gave you percent change in mass, so if I gave you a mystery solution, and if it gained mass, whatever this number is, then I could go, oh, the molarity must have been 0.28. Do you see how I would do that? Based on the trend that I got. In fact, I could have given you the same solutions unlabeled. I could have just colored them different colors. And I would say, hey, I gave you water. I gave you a 0.2, a 0.4, a 0.6, a 0.8. Here are the different colors. Find out which color is which molarity. Could you have done that? Yeah. You would put a little bit of the solution in the dialysis tubing. You would have waited initially. You would have waited after 30 minutes. And then you would have lined up the colors by who gained the most mass. Do you agree with that? And then you could tell me, this one gained the most mass. It must have been 1 molar. This one gained nothing or very little. It must have been what? 0. Okay? Ask the person next to you, do you understand the words coming out of her mouth? Because this is an application of the lab, and it is, in fact, one of your essay questions that I told you in your list of essay questions. So check with the person next to you and see if you understand that. Do you understand? Now, this is your second test. If you don't know where the essays are, you should probably check them out, okay? Because I've talked about it and I've told you how to study for it, right? So those resources are located on your APBio Google site, yes? In a folder called Resources for Review. In there where it says potential essay questions I could possibly ask, and I just compiled a bunch of different essays. And I told you don't look, one of them is old school essays, but they're still good to ask you. And those are separated by unit, and it's a PDF. Don't look at the unit numbers because they don't match the current units because everything's turned around now. Okay? But the other one is just lists of possible essays. And they go in about the sequence we're moving through, so just look at them and say, oh yeah, she could ask us that. Okay, the next part of the lab, I actually have a different slide. I actually have a different slide for that. Okay, the water potential. So, and you did a lab on this as well. So the prequel for understanding that is knowing that your water potential, why are we even talking about water potential? Why do we even have to talk about it at all? Why is this different from osmosis and everything else we've learned? Oh, you're making me cry. All your testosterone is dropping. Plants. Why? What do plants have that we don't? Cell walls. And what can cell walls do? Push back. Cell walls can push back. So when you talk about water potential, okay, you can calculate water potential for any solution. That's fine. But why this even comes up is because plants can push back, right, with their cell walls. So when you talk about plants, you say that water potential is a factor of two things. Pressure potential, which has to do with the wall, and solute potential, which has to do with osmotically active substances like sugar and salt. And you have to use both of this information in order to calculate the water potential. Because you could have a cell, a plant cell, let's say you take a carrot and you leave it outside all day tomorrow, and I think it's supposed to be smoking hot tomorrow. What's going to happen to that carrot? It's going to lose what? Water. Because the carrot will be what? Hypotonic, and the air around you will be what? Yeah, when you talk about the amount of water, not stuff, but pure water, there's going to be more water in that carrot than in the dry air around it, yeah? So it's going to lose water to its environment. It's all dry and shrivelly. So it is very hypertonic at this point, yes? I could put it in some distilled water. What's going to happen to the mass of that carrot? It's going to go back up because the water now, the distilled water around it would be what? Hypotonic, and the carrot would be? And water must flow from the hypo. Is enough water going to come into that carrot that makes it isotonic? No. Okay? Because it's pure distilled water out there and there's always going to be stuff inside that carrot. But will enough water come in that there's no net change in those carrot cells anymore? Yes. Why? Because the cell wall is going to what? Push back. Got that? So the carrot will still be hypertonic to the distilled water, but the wall surrounding the cells will be pushing water back out as fast as water is coming in, so there is no net change. Now, could I create a solution that is isotonic to the carrot? Yes. And if I create a solution that is isotonic to the carrot, will the wall have to push back? No. So then the pressure potential will be what? 0. And the water potential will in fact equal the solute potential. And that is what you discovered in part 3 of your lab. Right? Because you took chunks of potato and you put your chunks of potato in your different cups, all the way from 0 molarity, 0.2, 0.4, 0.6, 0.8, and what? 0.1. And you put your chunk of potato in there. You weighed your chunks of potato, your 3 cylinders, before. Then you left them in there for 24 hours and you weighed them after. And you did calculated the percent change in mass, yes? And what happened to the potatoes in the, oh, it's not going to work the same. Sorry. My bad. Okay, stop. Sorry. I'm not in Notebook. I'm in Google Slides. One more? Okay, here we go. I don't know how to not touch it. Okay, so when we did that, the potatoes in the 0 molarity compared to the potatoes, water must flow from the hypo, came into the potato got bigger. In 0.2 molarity, did it still get bigger? But not as big, right? And 0.4, what did it start to do? Got less, 0.6 more, 0.8 and 1.0, yes? So then we had a graph. Okay, we made a graph. And here was 0, here was gaining weight, here was losing weight, and we had our plot points or whatever, and then we made our best made line through those plot points, and we knew it sometimes it gained and sometimes it lost, and then we went down from that line and we looked to see what was the molarity at which it doesn't gain or lose weight. None of the molarities were exactly on. None of the molarities would be as I would describe as isotonic. But based on this, using our graph and what our other plot points were, we were able to identify what would be an isotonic solution to those particular potatoes, yes? And at that point, they would neither gain nor lose weight. So at that point, we could calculate the water potential by calculating just what the solute potential was. And we did that by negative ICRT. The negative is just because anytime you put something in water, the water potential goes negative. What is the I? Ionization. What did we use in our lab? Glucose. Does glucose ionize? No. So it's ionization. It's just one thing. So we just put a 1 here. The C was the molarity right here at which it would not gain or lose weight based on our predictions of what it did in 0.2, 0.4, 0.6. So I don't remember what we figured that out to be. Was it 0.3 something, right? I don't know what it was for you. Let's just say it's 0.3 molar, which is moles per liter, right? And then the R is just a constant, right? Liters per bar per moles per Kelvin. And then T is temperature, which you need to remember whatever temperature I give you to add it to 273 because it needs to be in Kelvin, right? And so then once we had that, sorry, this is so messy. And let's say it was, if we did, let's say 26 degrees Celsius. This is 99299. That would be 299 Kelvin. Then all I have to do is cancel out liters, moles, Kelvin, and I would And does anybody have a calculator so they can figure out for this particular molarity what it is? So we can apply it to a new situation? Do the math. Hurry up. Do the math at home too. My calculator has no juice. Anybody got it yet?
Yeah, Miss Lane, I have it.
You have it? Give it to me.
Negative 7.5.
Negative 7.5 bars. Yes? Okay. That is the water potential we would calculate based on that data of the potato. If I put that potato in a solution who had the water potential of minus 10 bars, what would happen to the potato? If I put this potato, we calculated, this is the potato, okay? Water potential. If I put that potato into a beaker whose water potential equals negative 10 bars, what would happen to the potato? Grow or shrivel? Shrivel, because it's closer to 0 than minus 10, yeah? If we think of it like a number line, right? The best possible water you could ever have, best possible water potential is always what? 0. So the closer I am to 0, the more water potential I have. So negative 7.5 is closer to 0 than minus 10. So it has a higher water potential. Water must flow from the hypo would shrivel. And conversely, then you can see if I put this in a solution who had minus 5 bars as its water potential, then the potato should what? Grow, right? Because it has a lower water potential. Minus 7.5 is lower than minus 5. So minus 5 would have a higher water potential. It would move into the potato. Turn toward your bio buddy and say, do you have that down, or is this something you're going to need to review? Now, I recorded for Mateen and Gosia, I recorded a whole thing on just that. So if there's parts of the presentation you know, fast forward. Here. This is what I need to review. And you can see me going over that with your lab. I did the whole thing already, right? If you needed more help about cell membrane, endocytosis, exocytosis, go find the video and just fast forward to that point. Does that make sense to you? Okay, next thing. Okay. All right, so this is the new stuff. All right, so the first thing I would say is endocrine system versus nervous system. Who's faster? Nervous. What does nervous use to transmit? Neurotransmitter. Neurotransmitter, where the endocrine system is slower and uses what? Hormones. What? Hormones, good. And what does the endocrine system use? Sort of the G. Glands, good. What does the nervous system use? Neurons, nerve cells, good. Which one has long-term effects? Glands, right? Unless you use your legs to jump off a building, and that will be long-term. Nervous system is usually for short, okay? All right. Structure and function. What I mean by that is you want to talk about how the whole thing works, the function of it. So an endocrine gland can release a what? Hormone. But it only affects the target tissues. How can the endocrine system be assured that only the target tissues will be impacted by that hormone? Yes, the target tissues have receptors. Then we could also talk about protein versus steroid right there, because protein hormones, their receptor is on the outside, right? Because proteins are water-soluble hormones, and most of our hormones are water-soluble proteins. And the hormone there is the first messenger, and then whatever happens inside is the second messenger. We looked at a couple examples of second messengers. We looked at cyclic AMP, remember that? And then calcium was the second messenger. Whereas steroid hormones, they don't stand at the door and knock. Steroid hormones come on in because they're made out of steroids, and lipids dissolve into lipids. So they can go right through the phospholipid bilayer and bind to inside the cell, or even all the way in the nucleus, and they affect the transcription and translation, right, of building proteins. That's why steroid hormones that are found in your ovaries, your testes, your adrenal cortex have long-term effects. Okay, whereas your protein hormones are usually very quick or rapid responses, like adrenaline, fight or flight right now. Whereas mineral corticoids and glucocorticoids and cortical sex hormones, those are long-term stress. Remember I gave the example AP over the course of a semester kind of stress. Not fight or flight right now in this moment, I'm taking a quiz kind of stress. All right? Then mechanisms of control. We said there were two kinds of feedback that hormones might use. One of those was positive, but that's pretty what? Rare. There weren't a lot of examples I gave you. The one I did give you is about what? Childbirth. And oxytocin makes the uterus contracts, pushes against the cervix, which sends a signal back to your brain to secrete more oxytocin. And this is going to repeat until you get a climactic event like a baby coming out, which really brings you back into homeostasis because the baby is out and you're on recovery. Yes? Okay? Negative feedback is more commonly used in our BioBuddy. And it's not negative like you're bad, right? But negative like it's self-correcting to that point where I'm trying to hover around. There's like a blood glucose level that is appropriate for your blood. If it gets too high, we're going to secrete what? Insulin. If it gets too low, we're going to secrete glucagon, right? If your body temperature is too hot, you're going to sweat. If you're too cold, you're going to shiver. Those are negative feedback. Okay? Now, actually, insulin and glucagon I would use as contrary hormones, right? That would be another way it's controlled. Okay, next, your highly suggested reading and thinking. Please don't miss them on this test. You know that I'm going to ask you about them, so please get it straight. And don't give me a bunch of baloney that you want me to read because I just get annoyed, okay? You know the questions, understand them, be ready to respond to them. Like I said, chapters 4 and 5, know your highly suggested reading and thinking. You do not need to do it for 40 yet, which is the stuff we learned today. You don't need to know those highly suggested reading and thinking. The other thing I don't want you to miss is your step further. Now, you know what you wrote on last time, and I told you should write it down so you keep track, because I keep track of everything that you wrote on, okay? So pick, if you did something on sustainability last time, pick NGSS, careers, or interesting information this time. So it needs to be different than what you wrote on last time. Now it's going to look exactly like it looked last time. First step, second step. So I would expect in first step, if you did the lawn grass probably isn't greener. If you read that, tell me, interesting information. You'll check the interesting information. Remember how you did that before? You'll check the box. You'll tell me about the long grass probably isn't greener. Just enough so I know that you read it. Your second step is tell me where you went from that. Tell me, I followed a link on that talked about blah, and this is what I learned about it. And just tell me a little bit about it. Okay? That is a gimme, right? Those are points you shall not miss, right? Because I'm telling you exactly what it's going to be. That and your highly suggested reading, and then plus those essays, right? And then the other thing you're going to do is you're going to work on your multiple choice. And you are going to start today. Do one thing today. I know it's Friday and you're tired. I'm going to try to grade one set of labs from one class, one group. I'm tired. Hope it's you. Okay? And so I'm going to try to get one thing done so that I'm more likely to get more done, right? Do something to help yourself prepare. Because you have Friday night, Saturday night, Sunday night, Monday night to get ready for this test, right? So do a little bit every day, okay? I believe in you. I need all the lab chairs up. So anywhere near a lab, please put up a lab chair. You're awesome. Make good choices. I love you.